Hello, this is Miss Dagenford again, and in this video lesson, I wanted us to learn about how the cell keeps its boundaries. And I want you to be thinking about what cells need to get in and what cells need to get out of them. So think about what we do as a human when we breathe in. What are we breathing in? And then when we breathe out, what are we breathing out? And that will give you a clue. So, and that's going to be different for plant cells, remember. And what do plant cells need? And what do plant cells need to get rid of? Now, so in this video lesson, we're going to be talking about that cell membrane. What in the world are these membranes that I keep referring to, what the book keeps referring to? Now, you might be wondering why I have a bottle of salad dressing, but this bottle of salad dressing is going to be really helpful to frame our understanding. We've all probably gone through that experience of taking the bottle of salad dressing out of the refrigerator, shaking it up, and setting it down, only to come back 10 minutes later and realizing that it's separated again into that oil and vinegar. And this goes back to our chemistry that we learned a couple of weeks ago. And I know you might be thinking, what? I thought we were done with the chemistry. Well, we're almost. And so, what we're going to learn about are these membranes of these cells. And you will be surprised to learn that, in essence, what's keeping us together, what keeps us from falling apart, are these membranes. And in essence, we are just fluid bounded by more fluid. And that's just astonishing to hear. I hope it's astonishing to hear. And then let's try to figure this out. Okay, so think about what job a membrane has to do. The outer membrane of a cell needs to keep the internal environment separate from the external. And that might be a little bit of a obvious thing, right? But then the membrane also has to separate those materials, but then allow some things in and some things out. Now, if you thought about it, if all of our cells need to get oxygen inside, and then all of our cells need to get carbon dioxide outside. And if it's a plant cell, it needs to do the opposite. Plants need to absorb carbon dioxide, and then plant cells need to get rid of oxygen. So, how is that happening? And then what do we even do about the food that we eat? How do our cells get sugars? How do our cells get amino acids? How do our cells get vitamins? So that's what we want to think about today. So before we move on, I want you to have three things firmly fixed in your mind. From a chemical standpoint, we need to revisit the polarity and non-polarity idea. Now remember, polar molecules are any molecules that have a partial positive and a partial negative side. So water, remember, was a really good example of a partial molecule. The hydrogens are partially positive and the oxygens are partially negative. And that meant that they were attracted to each other because of the partial negatives and partial positives. Now, that's also true for any other molecule that has polarity or a charge. So, this chlorine ion is going to be attracted to the positive hydrogen ion. And so, anything with a charge or that's polar is going to mix with anything else that has a charge or that's polar. So, polar mixes with polar. That's number one. Number two, polar will not mix with anything that's nonpolar. Nonpolar molecules did not have charges, they were neutral. So, number two is that polar and nonpolar do not mix. Oil and vinegar do not mix. And then nonpolar mixes with other nonpolar. So, like mixes with like. And you can come up with other examples besides salad dressing. Makeup and skin treatments. If you're going to use an oil base, then anything you put in needs to mix. And so a lot of this chemistry has a role. So let's go ahead and introduce our major player to the membrane. And it's this humble little molecule, and it's a phospholipid molecule. That's its name. Now, we mentioned these before, but very briefly. A phospholipid molecule should remind you of those large fat molecules that we learned about. And so we have these two long fatty acid chains. Now, these two long fatty acid chains are going to be nonpolar. And as they are nonpolar, they are also called hydrophobic. And the hydrophobic means that it will not mix with water. So these long fatty acid chains are nonpolar. Now, before you worry too much, I'm really going to show you this diagram right here, but really to get you to this cartoonish diagram. This cartoonish diagram down here in the bottom right is really the one that you need to be able to label and know. But I found in the past that if I only give the cartoonish diagram, students are just left with this, what is that? Like, you drew a circle and two squiggly lines. Like, what is that thing? So I always want to give you at least what the molecule looks like and then go to the cartoon. So we have this phospholipid molecule that has two nonpolar tails. Remember, nonpolar will not mix with water. And then up top, you'll have a part of it that does mix with water. It's polar. It has a negative charge here and a positive charge up top. And so this head is polar. It mixes with water. We call that hydrophilic, water loving, and then hydrophobic, water fearing. So while we talk about oil and vinegar not mixing, here's a molecule that has both a water loving part and an oil loving part to it, and water fearing part to it. So that ends up creating some interesting chemical behavior to it. So I'm going to show you this slide first. I'm going to go back to that other one. So this is slide 29. So we have a bunch of these phospholipid molecules and a beaker of water. And this beaker of water is going to, I'm going to put these phospholipid molecules into this beaker of water. And as I do that, and you can do the same with yours. Now, mine have already kind of been moved around, but you can, I'll have you pause in just a second, and then you can move yours around. Now, remember, these tails are nonpolar. And if these tails are nonpolar, they're not going to mix with water. And what ends up happening is they end up orienting themselves, arranging themselves. And of course, they're molecules. So when I mean they do this because they won't mix. So these phospholipid molecules will suddenly mix in a way that creates this little bubble where the nonpolar tails are in the middle, as close to each other as possible, and then excluding the water. And I got mine all mixed up there. Let me go back. There we go. And so, as much as possible, the tails are sticking to each other and mixing with each other, not the water. Now, at this point, pause and you go ahead and make two of these, and then we'll move on. So go ahead and pause the video. And this is what you should have. Two little bundles of these phospholipid molecules. Now, that's what happens if you have a bunch of them in water. But what if you had billions of phospholipid molecules in a cell? Well, because you have water on the inside of the cell and water on the outside, it ends up making a bilayer. It has it has two layers. But notice that the same holds true. The nonpolar tails are facing each other and away from water. So you'd have the watery inside and then the watery outside, but then you'd have these nonpolar tails creating this oily fluid. And it is a fluid. These phospholipid molecules are all jostling each other. It's not a rigid structure. So that's what I mean when I say that we are kind of kept together by a fluid surrounding another fluid. This is basically a layer of oil surrounding the cell. Embedded in these phospholipid molecules are membrane proteins that do different jobs. And notice the inside of the membrane that's mixing with nonpolar tails, that internal part of the protein needs to be nonpolar as well. So there are pictures. So we have different membrane proteins, some of which are receptors, some are enzymes, some form channels, some identify the cell, and some connect other cells to them. Now, one last thing we need to establish, and that is that depending on the chemistry of the molecule, some molecules can actually go right through the cell membrane. And that goes back to our three things. Remember, the tails, which are much longer, the tails are nonpolar. And those tails govern what can go in and out. If a molecule is nonpolar, it can mix with those nonpolar tails. And then if a molecule is small, it can then go faster. So the things to take away from this slide is if a molecule is very small, or if it's very hydrophobic or nonpolar, then it can just mix right through those nonpolar tails. So oxygen, carbon dioxide can go right through the cell membrane into our cells or outside of our cells. However, if a molecule is large or hydrophilic or polar or charged, then it's not going to be able to mix with those nonpolar tails. And so I've got some practice problems for you to do. So that's going to conclude our lesson here. And then we're going to go to, well, how do then cells get other things in?